So my name is Bernie Siegel, as you know, and I'm the founder and executive director of Genetics Policy Institute, founder of the World Stem Cell Summit, and thank you for attending our sessions, and I hope you enjoyed the meeting so far. I just want to mention that this is one of the sessions that is being recorded, and in about 2 weeks these will be available on the internet through the World Stem Cell Summit website. So this is going to attract a very large audience. And we are really honored, of course, to have Dr. Mahendra Rao, joining us for our session. So I just wanted to let Dr. Rao know how much I appreciate him coming. And I want to introduce my colleague, Rosario Sassi, a member of the Legal Advisory Board of the Genetics Policy Institute, and a professor and a colleague that has worked many years, including all the way back in the United Nations, back in the day in about 2003, 2004. She's with McGill University. Rosie?
Thank you. It's my pleasure to introduce a good colleague and friend, Dr. Mahendra Rao. As you know, Mahendra was the founding director of NIH Center for Regenerative Medicine and also the chief of laboratory of stem cell biology at NIH. Among many accomplishments, Dr. Rao laboratories focus on translational work related to Parkinson's disease and glial cell pathology. Dr. Rao is currently the VP of Strategic Affairs at Therapeutics and also serves as a consultant of regenerative medicine for the New York Stem Cell Foundation. Dr. Rao continues to provide consultant and advisory service to biotechnology companies such as Wscs, Megacorrion, Therapeutics, and Hemacare. Dr. Rao was recently named one of the top 10 influential people in the stem cell field and was honored recently by the Federation of Biologists, FABA, in India, for his achievements in the stem cell field and awarded an NBRI medal in India for his contributions to neuroscience research. Please welcome Dr. Rao.
Thank you. And I know it's an afternoon session, and I'll try my best not to put you all to sleep, right? But what I'm going to try and do is follow on for those of you who were here for the previous session, and sort of build on that to say, look, there's a lot of exciting work that can be done with IPSC and ESC, and here's how we have tried to reduce it to practice, but not just describe details, but make sure that these things are available to everybody in some fashion. So if I can have the slide deck up, please. So I'm going to focus, there are two sort of broad uses of pluripotent stem cells. One can imagine using pluripotent stem cells for cell therapy, and it's a whole separate large topic, and I won't talk about that in this session. And the other is to use cells for screening and drug discovery and for toxicology. And that's what I'm going to focus on in this. So I have a second title for my talk, and this was trying to do more with less. As you know, we've always had budget issues, and it's going to look like this is going to continue for the next few years. And so you have to try and plan on being innovative in being able to get things done without having to spend as much money as one would have usually spent in the past. Okay. So, quick start. Two ways we can think of using iPS cells or ES cells for regenerative medicine, and you heard about this from the previous speakers, so I'm going to go quickly through the first set of slides. And the one thing I want you to remember is two things, two important sort of properties of iPS cells. So iPS cells represent a paradigm shift in the field. And the reason they represent a paradigm shift is now we can get cells relatively easily. They aren't better than ES cells, they aren't different from ES cells, but I can now get iPS cells in a relatively easy fashion from every individual, pretty much, right? Even it's a 70-year-old or an 80-year-old, I can still get an iPS cell from it. That very fact that now, instead of thinking about one pluripotent line, but using large numbers of these lines, allows us to do certain things which were simply not possible before. And I'll try and give you some examples of that. So iPS cells can do everything that an ES cell can do, but one advantage that iPS cells have is that we can now do it prospectively, and we can make really large numbers of them, so we can do things with panels that we could not do with single cell lines. Okay. However, that doesn't mean that they're perfect paradigm, right? And there are problems with doing or using pluripotent cells for screening. And I want to highlight some of them here so that you don't think that this is a panacea, right? The first most important thing to remember is that humans take a long time to develop. And so that when you have cells in culture, they're also going to take a correspondingly longer time to develop. And it may be faster than what happens in normal biological development, but it's still going to be relatively slow compared to what happens with other cells that you put in culture. So on average, when you take cells and you put them in culture, it'll be difficult to model things which happen very late in development unless you can perform certain tricks in culture. Any of those tricks might change the actual properties that you're looking at, but it may not be the best model. So iPS cell-derived products are more immature even than if you were to harvest cells from fetal tissue and use them. And certainly they're going to be a lot more immature than taking adult cells and putting them directly in culture. So our rule of thumb for all screening is, if you can get cells from an adult and you can get them in sufficient numbers to do screening, then you should use an adult cell, because it'll be a lot more mature and it may reflect the disease in a reasonable fashion. On the other hand, many, many times we don't have a source of adult cells, or we can't get them in sufficient numbers, they undergo senescence, or we can't get the right kind of panels of cells. And in that case, you would use an IPSC derived product. So it's complementary, but you know which choices you're going to make when you do this. So the other big problem with using iPS cells for screening is simply expense. And I want to just give you some numbers from what's current technology right now. And the current technology says that it takes us about 6 months to make an iPS line. Yeah, you can do it in a shorter period of time, but you have to grow it up, you have to get sufficient numbers, and you have to characterize the cells, and that all takes time. So 6 months is a reasonable number of time before you can get an iPS line. It costs you about $10,000 just to make a line. And this is just an iPS line. It's not the differentiated cells that you actually want to use when you do any kind of screening. And it's one line. So if you're going to say that there's this advantage of taking iPS cells and using them for screening, you have to make a panel of lines. So it's starting to become pretty expensive. And then the really important thing to remember is that throughout all of the screening work that pharma companies do, etc., you have to remember they never used panels of lines. They used a single line. They used an immortalized line. It had to be cheap so that they could do high throughput screening. So this is going to be a big problem. It represents a paradigm shift. It represents an advantage that you can get cells you couldn't get before. You can get them to be certainly mature enough to do screening. Doesn't mean that they are perfect. And you still have to worry about the cost. Okay. So one big goal then is to see, can we reduce the cost in some fashion? So if instead of you making a line each time yourself, if there was a way you could access those lines, it would reduce your cost, right? Simply imagine if somebody else has spent the $50,000 it took to characterize the line, differentiate it, make some sufficient cells, and now you could buy it for $1,000. It's certainly much cheaper than having to do it yourself. And if there was a way that you could get them all relatively easily, then that would be a good thing to do. And so one of the first things I tried to do was to see if we could do this sort of thing and do it in a reasonable way and make sure that it was accessible. And I called it crowdsourcing. And that's just based on what people had done. And if you look up crowdsourcing on the internet, you'll know what I mean by that. So in general, we thought academia and pharma need sourcing and culturing of cells in appropriate numbers. They need to have these cells for screening where they have reporter lines, and they have a selection criteria so that they can make relatively purified populations of cells, some way to either sort them, etc. And then they would need access to panels of these lines, and that could be maintained in stock so that they were well characterized, so you didn't have to spend a lot of time trying to do quality control yourself each time to be able to do it. And then you would need not just the undifferentiated cells, because I'd really like to just do the screen with differentiated cells, so I'd need access to the differentiated cells. And I'd need to be able to make sure that I had some expertise so that I could do the right type of assays, because assays themselves require a certain set of expertise. It's not just let's look at the cells to be able to do it. So this is where we decided that we'll try and solve this problem in a crowdsourcing fashion. So I'm going to ask you to ignore the bottom slide. That's just there for me. Bottom part of the slide is just there for me to remind myself to tell you that this idea of crowdsourcing doesn't have to be only for drug discovery or toxicology or screening. It could also be applied to cell therapy, but I won't talk about that today. So our plan was very simply this. We said the government funds research grants for people to make iPS lines. If we could just make sure they could all be deposited in some kind of repository, and that they could be distributed to everybody else, you'd already be one step forward in having panels of lines which would automatically accumulate. If the government or somebody, non-profit foundations, etc., paid to get the characterization done on those lines, then that would be a really important way that you'd have quality control. And then if you had a non-profit repository, which could then take those cells and make sure that they were available widely, and they could be distributed both to academia and to non-academic centers which were trying to do the same work, so that you wouldn't have a cost or a transition cost in going from academia to pharma, then that would save time. And then they could then be able to do that. And then there's this one line here, and I want to emphasize this piece really, which I think is very important. When you screen for a particular function, whether it's a particular type of line, which is a particular property, but you may want to put in a reporter or knock out a second gene because you're looking at those things. And I'm going to call all of those things secondary products. So if you have a secondary product that you've made, there should be a mechanism that you could redeposit it back in a repository, so that now you've enhanced the value of the repository. So if there was a way that you could make sure that that happened, then you would be able to increase the size of the repository, and it would grow organically, and everybody would have the advantage of things that had been made. So you can imagine my lab may have gone in and taken a line which was there, the disease line, and now I put in a GFP reporter in it. If I can deposit that back in the repository, then nobody else has to make the same GFP reporter. They can now just use it from the repository. So that was sort of the idea of thinking about secondary products, et cetera, and being able to do it. So I had one advantage when I was thinking about all of this was that, boy, I work for the government. This was a time when I was working for the government. So the NIH can take a lead in being able to do that. And the intramural program certainly can take the lead in being able to set this as an example to be able to do it. There's one other thing that's really important in all of this that one needs to consider when one is thinking about this sort of crowdsourcing. The second part of crowdsourcing is always being able to share data, right? If you collect the data in the same cell line as a control, then you can compare across different lines, and you don't have to then always have to get two different lines and compare them yourself, or when somebody has a result in a particular line and they're across the world, you don't have to go back and talk to them directly as long as they've used a common control. So the second part of making this sort of crowdsourcing possible was to have some kind of calibration line or reference material. Nothing had to be very good or bad, but it just had to be consistent, that was widely available so that people could use it and generate a lot of data in one line, and that would be akin to what pharma had been doing in the past. Everybody did that work in either oocytes, or they did in HEC293 cells, or they did it in PC12 cells if they were thinking about Parkinson's or neurons. And all that data was available from one pharma company to the others, which was published. So I knew that I could compare my data, or I knew what would be different if I did work with one drug versus another drug. So we thought that that could also be applied here. So that gave us two goals. Make sure that there's a reference material that's available, and that should be widely available, and it should be well characterized. And the other is to make sure that there would be large numbers of lines which would be available through the repository at a reasonable cost. Okay. So I'll go through this very quickly and say it all worked. And the way we did it was we worked with 3 or 4 of the nonprofit repositories, and I'd skip this slide in terms of being able to do it. And we arranged that the NIH itself, as part of jumpstarting the program, would make at least 10 or 12 what would be called reference or characterization lines or calibration lines available. They were called NIH CRM 126. We asked Jamie Thompson's lab, because they were well known for their expertise in making lines, to contribute a couple of lines, and they did. We asked a company called CDI to contribute a line because they were making that commercially available to a lot of people, and they did. We asked Dr. Shinya Yamanaka's group to contribute lines, and they did. And so that we'd have a representative set of lines, and these lines would be all integration-free. They'd all be well characterized. They'd have whole genome sequence, and they would have microarray analysis data and epigenome data available. More importantly, they'd be all available from one single repository. And they would be available both to academia and to industry. And the data would be available as well. So now you would have a way to be able to easily compare or contrast, and you could take a line that you wanted to use. We decided that of the lines we chose, all of them should be integration-free, to allow you to be able to assess off-target effects, and that they would be all adapted to the same media in the repository, so they were all in the same media. And more importantly, we decided that one important comparison which needed to be made was that male lines should be available as well as female lines, and so there are both male and female lines that are available, made from the same source or a different source in being able to do it. So that was the first step, and that got done. Then the second step was to say, look, if we're doing this for IPSC, we should also have something like this for ESC. Luckily for ESC, it was already done in some fashion thanks to Wscs and Worf, but we wanted to make sure that those lines were available and they were well characterized and that there'd be some correspondence between them. So we said that we needed to do one additional thing, and that was to make IPSC from differentiated cells from ESC. So we took an ESC line, and we took the H9 line, and we took the H14 line, and we differentiated them into a differentiated cell product. In this case, it was neural stem cells. And we took those neural stem cells and we reprogrammed them to make IPSC. So same isogenic background, same genetic material, but now you had an ESC line and you had an IPSC line, and you could compare them. We actually were even more paranoid about controls. So what we decided to do was one more thing. We made them with integration methods, because a lot of people were doing this with integration methods, and we made them with integration-free methods. And we said those lines will be available to anybody who wants to use them. Okay. The next thing we said was that, well, what about other lines, right? So now you've got controls, but you really want to do experiments. So can we make other lines available? And these are patient-specific lines or disease-specific lines. And we could take advantage of the fact that the NIH is the world's largest clinical research center, and it's famous for having the largest access to orphan and rare diseases. So we asked our colleagues at NIH that would they contribute lines from all of these rare orphan disease patients, and we'll all put them in a repository. So I'm here today to tell you that it's not all happened yet, but the process is in place, and we estimate that there are about 450 lines that have been made by the intramural program at the NIH, of which we are starting to make sure that they're deposited. And this includes all the major orphan and rare disease lines, diseases. So these are the different repositories that we've been working with, and I just want to highlight one repository here, which is the one at the bottom here, and that's Sigma. And the reason for Sigma is that Sigma is not a non-profit. In fact, it's a for-profit, and it's not really a repository, right? However, there was one huge plus on why we thought having Sigma sign on to all of this was going to be important, and that is because Sigma controlled the technology with zinc fingers and gene editing. And you heard a lot about gene editing in a couple of sessions earlier, so I won't belabor it, but what it meant was that there was a potential that lines which had been engineered using zinc fingers and talons, because talons there was an open source system, it would be possible to include them in such a repository. And so we spent a lot of time and effort with Sigma to get them to agree that zinc finger engineered lines could be deposited in a similar repository and would be widely available for people. So that was quite a success. So we were pretty pleased with this. And the stage so far is that we've got at least all the reference lines in place. We have about 50 or so of the disease-specific lines already deposited, and we have about 10 or 15 of these gene-engineered lines which have already been deposited with Rutgers, and they're available to individuals, or they're in the process of being characterized, and they can be widely distributed. Okay. So that was great. We thought at least initial success in being able to do it. This is published. People had already started using it, and in fact, I got a report from Michael at Rutgers, and he said that more than 18 companies have been able to access these lines and use them in their internal purposes as well. So that's sort of nice. It says that the whole idea was validated. So the next question was, could we extend this not just to the undifferentiated iPS cell, but also to the differentiated products that come from them? And so we worked very hard with all the people who hold the right patents to make sure that this could be done, and we had two strategies in terms of what we could do with this. It turned out that the repositories themselves said they don't have all the expertise to make all the differentiated cells, and you heard in the earlier session why that's a difficult thing as well. So we said here we have to co-opt a couple of groups. One was the university packed centers. This was the Heart, Lung, and Blood Institute had funded a lot of people to be able to make differentiated cells, and they supported it through the NIH. So we said maybe they can help with providing differentiated cells. Another group was Darwin Prokop's group in Texas, and what he was willing to do was take iPS cells and make MSC from them and distribute MSC that had been made that way. And keep that thought in mind, I'll come back to it. And then we thought that if we could persuade companies to be able to distribute these lines, then that would be okay as well. And as a test case, we identified 3 companies which worked in the nervous system, and I'll tell you later about how we had developed protocols for making differentiated cells, and asked them if they thought that there was a business model for them to be able to provide differentiated cells. And this would include just normal differentiated cells, patient-specific differentiated cells, and engineered differentiated cells. And the answer was there were at least 3 or 4 companies which were willing to be able to do that. So now we had a model system where you could use this sort of crowdsourcing strategy, and you could combine it with being able to make sure that the cells were available to academics and non-academics, and would be available either through non-profit repositories or through for-profit repositories and companies, and the pricing would be relatively reasonable. And what I mean by being relatively reasonable is the following, right? If you want to buy an IPSC line, which has been well characterized with all its whole genome sequencing, et cetera, and want to get it from Rutgers, the price is roughly between $500 for 2 million cells to $1,000, $1,200, depending on whether it's a company or it's an academic to be able to get those cells. If you want to get a differentiated cell like a neuron or astrocyte, oligodendrocyte, because there's more work in differentiating them, it's in the range of about $1,000 for a couple of million cells. And a couple of million cells, I want you to remember, for screening is enough to do 1 96-well plate to 3 96-well plates. So that means you can screen 100 compounds in triplicate with this kind of $900 cost. So this is starting to become something that's feasible for pharma. And you heard in the previous session that pharma is beginning to start doing this. And so that's why I mentioned the number of companies, and sort of 18 companies have already started taking these cells and using them. Okay. So we've extended it to differentiated cells. We needed to extend it to engineered cells. And we've been able to work with Life Technology or Thermo Fisher now, and with Sigma and others, to make sure that the engineered cells could also be widely distributed. And I'll talk about this a little bit later. Okay. So here are some quick examples to tell you this is not all sort of pie in the sky, or in software as they call it vaporware, right? It's actually true, it's there. You can go to the websites of these different repositories and look at what cell lines are available, and there'll be a lot more going to be available in this coming year. So that's engineered lines, they're made with CDI, those are available. Here's engineering tools, so the zinc fingers that were used to make those lines are also available, so you don't have to just use the same engineered lines, you can make your own, which would then be identical to what is available as well. And we have about 200 such well-characterized zinc fingers that are now available. Okay. So now, this is all theoretical, right? I've told you, yes, companies are getting the lines, yes, IPS lines are available, but are they all useful at all, right? And so I'm going to give you some examples from work that we've done using these lines, and show you how quickly things can be there and how you can get them done. And I'm using what I have done in my lab because I ran a relatively small lab. And so if I can show you screening that a small lab can do at a cost that a small lab can afford, then it means that it's actually sort of worked, right? Okay. So the first simple thing was to say, can we screen with IPS lines? Can we use multiple IPS lines and do a simple standard screen? So here's a screen which we did with a standard compound library. It's called the Spectrum library. It's about 1,200 compounds. And I had one student work with me to be able to get this done. And this was pretty straightforward. You had IPS cells, you were growing them. It cost me $500 to get 2 million cells. I passaged them once, and now I had them so that I had more than 10 96-well plates, so I could screen 1,000, 1,200 compounds pretty easily, right? We did this in triplicate, then we identified a subset, we reran that subset, and our goal was very simple. We wanted to identify a compound which would kill IPS cells but would not kill differentiated cells. Because if we could do that, we would have a therapeutic candidate that could be used when we are manufacturing differentiated products from IPSC, right? And we could prove that the cells, that there would be no contaminating pluripotent cell present in the culture dish. And the nice thing was we found 6 classes of these compounds. And then we could mix and match them, and we could show that at a relatively low dose, we could use them. And because we had multiple lines, we could test them on 10 or 12 lines and show that this compound worked with all of those lines when we differentiated them into an appropriate phenotype. So that became a very simple thing. It's published, so I won't go through all the details on how we did it, but the answer was yes, it can be done. So certainly we could screen at the IPSC stage. Okay. Now this was a disadvantage, okay? At the IPSC stage, for most patient-specific lines, most familial diseases, etc., there is no difference. So you can't screen at the IPSC stage for a genetic defect which affects cells which are differentiated. So it's a good thing and a bad thing. It means I can make IPS lines, but it's also a bad thing is that I can't screen at the IPSC stage. So while I could show that I could screen at the IPSC stage, it was just a small thing, right? Most things I can't screen at the IPSC stage. I still have to differentiate them. So we decided, let's figure out a way that we can set up to be able to do a standardized differentiation protocol so that we could get all of the differentiated cells we need in the central nervous system and see whether we could run a screen. And the reason why we wanted to get all the cells was that we might want to do mix and match experiments, because in the nervous system that's important. The cells talk to each other. Maybe it's the defect in the astrocytes which is causing a defect in the normal neurons, or vice versa, and we'd be able to dissect this out relatively easily. And we wanted to make sure that we had a relatively pure population of the cells. And we had to make sure that it was cheap, right? And we wanted to make sure that the SOPs could be transferred to companies so that they could sell those differentiated cells. So we looked at what we could do. And the first test we thought might be really useful to do, and now this is commercially available, so I'm going to just say that we weren't the first, but other people did it, but we had started the process. Pfizer and others had introduced something which was called the embryoid bodies test for toxicology. You made embryoid bodies, and you could just screen toxicity of compounds by just looking at a single embryoid body in a dish, so that you had a sort of 96-well assay to be able to do it. So all we needed to be able to do that EST test and transfer it from mice to humans was to be able to take the cells and show that you can make consistent embryoid bodies, and that those embryoid bodies could be used in a reasonable way. And it turned out that the answer was yes. We could make very uniform embryoid bodies. Oops. Too many buttons. Okay. So we decided that we wanted to see whether this could be done. And the answer was yes, we could do a screen at the embryoid body stage. I'm not going to show you the whole screen. I'm just going to highlight a couple of differences between mice and human here. So in mice, we could do the embryoid body test at 4 days. So we made an embryoid body, and we got consistent cardiomyocyte differentiation, and that was an easy way to be able to do it. With humans, we needed to wait at least 12 days. And at 12 days, we did not get consistent cardiomyocyte differentiation. We did get ectoderm, endoderm, and mesoderm, so the assay was much longer. So we had to figure out whether there was another simple measure rather than using cardiomyocyte and beating embryoid bodies as an assay. And it turned out that there was a PCR-based readout that we could use in terms of differentiation and biasing things. So it was a multiplexed PCR result. But take-home message was, you can do a screen and you can mimic what people had done with mouse embryoid bodies to be able to run a screen that had been pioneered as an alternative to doing animal testing for toxicology. Okay. So next thing we decided was we needed to make neurons, astrocytes, and oligodendrocytes. And here is where we ran into one problem. And what this was our solution to that problem was the following. When we normally make neurons or astrocytes or oligodendrocytes, there were many ways, and it all worked, and there were many protocols. The problem was consistency, right? If I did it directly from an IPSC in a 96-well dish, then each well was different. If I did it and I tried to plate the finely differentiated cell, it was still not consistent because the plating for differentiated cells was difficult in being able to get that done. And if I just had to grow things with an intermediate and keep them for a long time in a 96-well or a 384-well, then feeding the cells started becoming a problem when I went to a 384 or a 1536-well plate. So all of these were sort of technical problems, but they meant that I could not really do a screen in a cost-effective way. So could I solve that problem? And the trick that came up, which worked for us, was to see if we could find an intermediate stage which was stable, where I could expand the cells and get sufficient number of cells. And it turned out to be something which I had worked on when I was a young professor in the University of Utah, which was we had identified the neural stem cell stage. This was a neural stem cell which was formed, it was called the neuroepithelial cell, which was at a stage earlier than what people had described when they talked about neurospheres, and that was an FGF-dependent cell. And long time ago, we had shown that we could make this from mouse ES cells. So we just went and checked whether we could do it from human, and the answer was yes. And so we could make neural stem cells, and we could grow the cells and expand them and keep them in culture. And you heard from the previous speaker, there are two different ways to do it. You can do it through embryoid bodies, or you can do it through direct differentiation. In each case, you get the cells that are really pretty good. More importantly, we could show the following thing, that this intermediate step, after we'd stored it and expanded it, we could still make neurons, astrocytes, and oligodendrocytes from them. And more importantly, we could make multiple kinds of neurons by directing the positional information that we expose the cells to. So we could make forebrain cells, or we could make spinal cord motor neurons from the same starting population. For people who do manufacturing, you'll see why this is a huge advantage, right? Common starting material you can store it at an intermediate stage, and you shorten the time before you put it into a 96-well plate, so you get a lot more consistency. So again, I don't want you to think this is theoretical. The reason I'm presenting it this way is to say that we actually tested this, okay, to make sure that this could all be done, and I'll show you some of those results really quickly. Okay. So we figured out a method to be able to do it. We figured out a way to store the NSC, and we confirmed that we could take the same starting population, we could make multiple kinds of neurons from them, as well as being able to make astrocytes and oligodendrocytes from that same starting population. There's a reason why I'm showing this is to make one very important point from some questions that came up in the last session. We were really lucky. It turned out that that this normal development that occurs in the brain is the following. You get neurons which are born first, followed by oligoprecursors, followed by astrocytes, and then you get oligodendrocytes later in development. And this is spread over almost a year of embryonic development and postnatal life, right? So that's a really important thing to remember. So we thought that that is what will happen here, and if that's the case, we'll be lucky that we should be able to get a relatively pure population of neurons, and if we kept the cells in culture for a longer time period, we'd get neurons plus astrocytes, and we kept them even longer, we'd get neurons, astrocytes, and oligodendrocytes. There's one other trick that we'd learned when we were students, which was that when you passage cells, then the post-mitotic neurons die. So that if you passage your astrocyte and neuron culture, you're going to get a pure population of astrocytes without having to do any sorting. We were trying to avoid sorting. We knew how to sort, but if we avoided it, it saved us a lot of time and money in being able to do it. And it turned out that you could make oligoprecursors if you changed the conditions correctly and you added inhibitors to making astrocytes. So we could now get rid of the neurons, we could inhibit the astrocyte formation, and we could get pure populations of oligoprecursors, all without sorting. We could sort, but we wanted to save that cost in being able to do that as well. Because in a separate part of my life, we'd also identified cell surface markers for each one of these things. Okay. And then I just want to show you that we could make dopaminergic neurons as well, and the reason I'm showing you this is just as a quick slide, because I'm going to show you a screen that we did with all of this. Okay. So the last piece that I want to tell you about all of this is one thing we learned from our pharma colleagues. They said, yeah, you can make neurons, you can make astrocytes, you can make oligodendrocytes, but when we run an assay, we want to be able to make sure that we can do it quickly. We might want to be able to put a reporter in it, and we might be able to make sure we can quantify in an easy way without having to actually count cells. So we wanted to look to see whether we could do gene engineering in these cells or put in a reporter either by transfection or by using lentivirus or some other possibility here. And all I want to show you here is that we can do all of those things here. So I just won't talk about this, but we can do multiple types of assays here. So we can look at live dead cells, we can look at metabolic activity of the cells, we can look at synaptogenesis with the cells, we can look at electrical activity with calcium imaging, and we can transfect in organelle reporters so that we can look at the state of the mitochondria or the state of the lysosome, etc., and being able to do it. And I'm not going to show you each one of those things, but trust me, once you have a large number of cells and you have them in reasonable number, it's not a difficult thing to do given what has already been done in the past. Okay? So the next few slides, I'm just going to show you a quick screen, examples of screens that we've run. And I've chosen 4 examples here of quick screens. And again, the details are not that important. What's important is that these screens should convince you of the following. It means we can do it, right? That's the first most important. The second thing is it means we must have been able to put things into 96 well plates or 384 well plates to be able to do it. And it means we must have relatively pure populations of cells to be able to get to the stage that we are doing it. Okay. So the first thing we wanted to do was a toxicology assay. This has been a big problem for neurotox in the field because it's been very hard to get neurons and neural stem cells, etc. And when we try to do them in vivo, we have the blood-brain barrier to contend with, so it's not been easy to do it, and it's been very expensive to do neurotox. So our hope was that we could get pure populations of neural stem cells, we could get neurons, we should get astrocytes, and we should be able to now look and ask a very simple question. The simple question would be, is the result with human cells the same as with mouse cells, which is what has been used as the state of the art? Is the result the same between neural stem cells, neurons, and astrocytes and oligodendrocytes? We don't expect it to be the case, but we should be able to find that out. And can we determine mechanism of action relatively quickly if we have this sort of high content screen available for us in a 96 well plate? So we use the Spectrum library. This is about 2,000 compounds. These are all the FDA-approved compounds, and we could just test them very quickly. We use an MTT viability assay to be able to do it, and we ran it in triplicate. And for the purposes of this talk, we focused on iPSC and neural stem cells, and we looked for things that would kill neural stem cells and kill iPSC or not kill one or the other. And so that was similar to what we'd done in the past, and so we knew that we had an internal control. And then we took those same compounds and we looked at them in neurons, astrocytes, and oligodendrocytes to ask, is there a difference in cell type behavior, etc. Quick answer is it worked. These were the hits that we found. We were able to look at them, and since we were doing this all in triplicate, we could then go back and do a dose with the positives and ask whether that was the case. And then we could go back and find a resolution, dose response curves with this. And I want to give you this one or two examples because I want to emphasize a couple of things here. So one of the things that killed neural stem cells was actually digoxin. And the important thing was that it killed it in a plasma concentration dose, which is consistent with what's used for people who are being treated for congestive cardiac failure in chronic ways. So this crosses the blood-brain barrier. So that suggested that this may be a specific thing. And it did not kill neurons and astrocytes. So this was specific to stem cells. So that was another important finding for us. It's really important as well, not going to show all the details, was that this did not kill mouse neural stem cells. So there was a species difference here. And it turns out, of course, this is when you learn that you're being stupid in some fashion or you don't know enough in a field when you're starting off first. It turns out people knew this. Okay? So when we read the literature, they said, mouse cells have a different receptor and digoxin doesn't bind to that class of receptors, but it binds to the human receptor. So we should have been able to predict it, but we didn't know this, so it was kind of gratifying to find it out in the literature after we'd done this. So this, on the bottom right, you'll see here that there's another figure here. So this is a figure from a reporter line. So we took the same neural stem cells and we put in a reporter, in this case it's a RFP reporter, so that we could visualize the cells and ask, during the process of dying, how do the cells die? Okay. And it's a simple way to be able to do it, and you can measure it, and this is just a volume ratio that you can map when you do it with standard software. I won't go through the details, but the important thing here was when digoxin killed the cells, it looked like the cells blew up. So they expanded and then they contracted. So that tells us that it's likely to be a transporter effect. And so we know that digoxin has multiple effects, but there is a candidate sodium transporter that it may be involved in. And so very quickly we were able to localize the transporter that was involved in this particular mechanism of toxicity. In fact, that was why we were able to figure out that it's a difference between mouse and human in being able to do that. So I want to pause here a minute and try and emphasize again what I've tried to say. Relatively cheap cells, you can put them into a 96-well plate, you can look at them by high content screening, and you can look at viability, and you can do it relatively quickly. A small lab can do it because you can take these cells and put 1 or 2 million cells at a relatively $900 cost and be able to get 100 compounds, right? So this whole screen can be done by a couple of students, and you can get this sort of result and relatively quickly get the mechanism of action. This would have been very, very difficult to do any other way, right? So now you can also imagine how you could take this to the next step, right, in being able to do these things. I can also now look at interactions between the drugs, because now I have drugs and I have a dosing for these things and I know what the mechanism of action is, so I can see if there's additive effects, synergistic effects, or they inhibit each other in terms of being able to do it. And we were able to show that there's a class of cardiac glycosides and they all act through the same mechanism so that they're not additive, but they inhibit each other's activity because they compete for binding in being able to do that. Okay. And lastly, I want to remind people of what I had started off with. So we found that there were drugs which were specific in killing rodent cells but not killing human cells, and vice versa. There were ones which were killing human cells but not killing rodent cells. The general rule of thumb seems to be that human cells are much more susceptible compared to rodent cells, which seem to be a little bit more resistant. So in all classes, if you looked at 2,000 compounds, we'd get a smaller number of hits when we used rodent cells, and we'd get a larger number of hits, but they wouldn't overlap, okay, in being able to do it. So somehow there's that kind of difference, and whether it's a culture artifact or whether it's real, we don't know, but that's what we found. Okay. So this tells us the importance of being able to compare data, and it tells us that we have the ability to compare data with historical rodent data and get the new human data so that we can move forward relatively quickly. Okay. So I'll skip this slide. It just reminds me to tell you people that this was how we could do this and that it's affordable and it's relatively straightforward in doing it, in being able to do that. I'm going to take the same example and now go to one more cell type. So this is, I showed you an example with toxicity screening and being able to look at a generic cell type. The second example I want to give you is to take a specific phenotype in a particular disease. In this case, it's Parkinson's disease. And in Parkinson's disease, there is a model that people have used for many years in trying to identify drugs, and that's called the MPTP, or the 6-hydroxydopa model, where you grow the cells in culture and you add either MPTP or 6-hydroxydopa and you can kill the cells. Long time ago, we'd shown that we had a positive control in that if we added GDNF to the cultures, we could prevent the MPTP toxicity. So we had a range. So we had an assay, right? And it was a live dead type assay, which we could use. So we did a very simple screen, and this is to mimic what would be a drug repurposing screen, and asked, these are the things that people have identified using rodent cells or in vivo rodent models of MPTP or 6-hydroxydopa. Can we now take this and see which of those drugs actually works with human cells? And there's a caveat to this experiment. If it didn't work with human cells, it could be because the assay is not great, or it didn't work because it's truly different between rodents and humans, right? So keep that in thought in mind. So here's the quick result. So we tested 50 compounds, 44 of them are shown here, and out of those 44 compounds, there were 20 which did not work in human cells. It's a pretty significant number, and the remaining did, right? So now we had a question. Were the ones that we found real, and the ones which we didn't not real, and how would we know? And the only way we know is by going back to the literature and asking whether clinical trials had been done in humans and which ones passed or failed. And the answer was that the majority that we found seemed to be the ones which seemed to have an in vivo effect, and others did not. We didn't have a one-to-one correlation in testing all of them, but it certainly was very encouraging, right? So what it told us in general terms was that using human cells is probably better than using rodent cells, and that there's going to be differences between human and rodent cells when you do these sorts of assays. And perhaps that is something that's important to start considering when you're considering different assays that you run. Okay. I want to show you two more small pieces about how we can extend the utility of the assay. So one thing I told you was high content screening. The other thing is we can now do whole genome expression analysis in a relatively cheap way to be able to do it. And we spent quite a lot of time making sure we could do whole genome expression analysis from cells in 196-well plate. Okay. And this was something that had been discovered with the Broad Institute, and we were able to use that as well with links, but now you can actually do it with a standard microarray. So we can look at all 32,000 transcripts that are possibly present, and we can look at it from 196-well assay, and the cost is reasonable in terms of being able to do it. And so we were able to use this to very rapidly understand the mechanism of toxicity or the mechanism of action of a drug, especially when we combined it with high content screening. So next piece of being able to use these cells for drug discovery is to say, well, you could do normal cells, you could do panels of cells, and you could look at particular disease models that you could do with small molecule compounds. Can you take it to the next step and be able to do it with disease samples, right? And we decided that, yes, we can. So we can go to patients and we can make patient-specific lines, and we would have those patient-specific lines in which we could do all of those things. So we've done this with several diseases. We've looked at ALS, we've looked at Parkinson's disease with PARC, null models, etc., and with several of the orphan and rare diseases. I'm going to very quickly show you two examples. So it's nothing novel, except the only difference is we can do a same sort of screen, except we can take a normal cell and we can take a patient-specific cell, and we can look at these things relatively quickly. So I'll skip this, and this is Niemann-Pick. So we made an IPS line from a Niemann-Pick type C model, and we made neurons from it, and we looked at neural stem cells, and we looked at astrocytes, and we looked at oligodendrocytes. And a student in my lab, anesthesia, made this observation, that when she grew Niemann-Pick cells in culture, the neurons died after 10 days in culture. The normal cells grew fine. Okay? And so that was great. We had an assay, right? And we had an assay without doing any additional perturbation. So now we could look at a screen to see which drugs prevented this death. Right? And that would be a candidate drug that we could possibly use. And again, because we are paranoid, we could now go in and say, this is what people have identified in the literature. Maybe that will work, but we can also look for new things and see what would protect in that same sort of assay. And so the paper's just been accepted, but what we were able to do was to be able to run that screen. And the good thing was that curcumin, which people had described before, and there were a couple of other drugs, that cyclodextrin, that people had described, when we could show that they worked, and that they worked in protecting. So that was kind of nice, right? We could show that. What was important was some of the drugs that worked here did not work in fibroblasts. So it told you had to have the right kind of phenotype in which to look at the cells. Neurons are the ones which are damaged in Niemann-Pick, rather than NSC or IPSC or fibroblasts, which is what people had used for screens before. But we could also use other drugs, and I won't go through the whole history of why we chose what we did. But the one thing we found, which was kind of exciting, was the Wnt family of drugs. So it turned out that a subclass of the Wnt mimics were very useful in protecting against Niemann-Pick neurons dying. And so we had a new class of drugs that we can now screen as lead compounds to be able to move forward for therapy. So that's been a sort of important early thing that we've been able to do. And I'll skip all of this because it's just data that we've done. And we've been able to do similar things with other diseases as well. And I won't go through all of that as well, except for talk about Gaucher's disease, if I have the slide here. Oh, I don't have the slide here. So we did the same thing for Gaucher's disease, where we were able to show that there's a drug that may be really useful as well. And in that case, that drug is now at the NIH going forward with the phase 1 trial, and I think 2 patients have been treated with that drug already. So you can see the power of the ability to be able to screen with pluripotent cells and making differentiated cells from them and being able to move this sort of thing forward. Right? So in the last section of my talk, I think I have time, a little bit of time here still. I want to talk about how you can expand the versatility of this sort of screening. Right? So remember, I've told you about 3 kinds of screening, right? You've got the right cell type in which you can screen. You can put in a reporter into those cells so that you can screen in a particular way or a particular organelle in case you want to do it. You can make a patient-specific line to be able to screen, and you can take panels of lines and be able to screen different stages of cells so that you can discover things. And you can combine it with high-content screening, or you can combine it with whole genome expression analysis to be able to rapidly get to the mechanism of action. So what more can we do with being able to get all of these things done? And I want you to go back, for those of you who were here, early in the morning when George Church talked about genome editing, right? So genome editing is a very powerful technique, and it's allowed us to be able to do a whole lot of things. And so I'm not going to talk about the whole therapy part of things and replacement, but for gene discovery or for doing drug screening, there are 2 important things you can do. One, you can put reporters into a defined site in the genome. When you do that, you avoid any of the off-target effects that you have with lentivirus and multiple copies and making sure that each time you get the same line or you don't get the line. The importance of having that defined locus or a safe harbor site is once it's well characterized, it doesn't matter if in 100 different lines I put in that same reporter, it'll likely behave the same way because it's targeted to the same site. So that's really important that I can use the same reporter. This has been a huge problem for screening. Every time you take a lentivirus and you put a reporter in, you have to now make sure that it gives you the same readout, it doesn't shut off, nothing happens to it because there are lots of other issues that are there and you have to pick clone. So this was sort of one thing that was going to be really important. The other thing was, if you have a patient-specific line, you can now make 2 mutants in that line. You can add a second mutant, so you can look at what is the effect of combinatorial things which you don't find in a disease. So that's a really easy thing that you can get in a screen. So in Parkinson's, there are 5 different genes which affect, which can cause you to get Parkinson's. So you can look at the effect of these 2 on a pathway and whether they're enhancer or suppressors of a disease. So that's a very simple thing that you could do in being able to get that sort of engineering done. And the third thing you can do is you can make isogenic controls. So you can go back and introduce the mutant in the same allelic background. And once you do that, then it's less likely that it's because of that combinatorial effect, but it's because of that particular gene, and it's because that is necessary and sufficient in causing a disease. So I don't have time to show you all of that, but I'm going to give you some examples to hopefully convince you that this is actually possible. So we decided that we wanted to make sure about the safe harbor locus and that we could put reporters into that safe harbor locus and that we could do it at high efficiency so that you could do this on a routine basis and you could build lines and reporters. So we asked ourselves, what do we really need? If we're looking at toxicology screens in the nervous system and we're looking at things, and when we talk to people, my colleagues said, we need some generic reporters. So a strong reporter so that if you do in vivo work or you're doing transplant work or you're doing preclinical studies or you're looking to quantify, you want to have some kind of reporter. And either GFP or luciferase would be really good if you could put that into a safe harbor. And if you could put exactly the same reporter but in a lineage fashion. So if you had a neuronal reporter or a glial reporter or oligo reporter, then that would be great. And we thought all of these things should be possible. There were two safe harbors that we had identified. One was in chromosome 19, the other one was in chromosome 13, and we'd published on those before. So if we could get a construct and we could show that that worked, then that would be great. We had to find a reporter that would be ideal, and we had to have the license for that, but we figured that out. And then if we had a neuronal marker, and we decided either MAP2 or beta-3 tubulin would be good markers for that, or double cotton, and for astrocytes, everybody said GFAP would be a great locus, and for oligos, myelin basic protein, which is expressed relatively early, even before the onset of myelination, would be a good marker to be able to do that. And then for a reporter, everybody said, we want to be able to quantify. So GFP is great, but it doesn't change rapidly. So when you're doing screens, we prefer something else. And so we thought luciferase might be a good candidate for that. Luciferase has two advantages. You can actually use a chromogen so that you can look at it by fluorescence, or you can actually do enzyme activity, which is quite sensitive and it's quite quantifiable. And we added a new wrinkle to it. We said we'll put an HA tag to which antibodies are available, so you can fix and stain as well. So we built that construct, and we looked at two safe harbors, and I'll skip all of these slides and being able to do it. And we found that we could make these reporters, and we could make them with relatively high efficiency in these two sites, either using zinc fingers or using talents. Okay. So we were able to do that. Okay. I'll skip this slide. Let's go back 1 minute. Okay. So, oops, it looks like. Okay. So, I think I did not put the whole list of markers or reporters we made, but the take-home message was we were able to do this. So now we have reporters for double cotton, a reporter for GFAP, and a reporter for myelin basic protein, and we have a ubiquitous reporter in the safe harbor site at chromosome 13, which expresses luciferase halotag constructs so that we can do this sort of screening. And all of them are in the same background, right? So we have the normal line, and you have all of these reporters in the same line. And you have constructs so that you can make identical reporters in your own disease-specific line so that you can do comparative expression analysis as well. Let's skip this and go to one last slide because of the previous talk here. And that was, why did we only do this with IPSC? And in my mind, there is no specific reason for using IPSC other than the fact that we can make panels relatively easily. And there are a large number of ESC lines also available. So we wanted to test whether we could do exactly the same thing with ESC lines as well. And the answer was yes, the efficiency is the same. And we were able to take the H9 and H14, which are available from Yscell in that bank, and make corresponding reporter lines with those as well. And we were lucky there that Sucheng Zhang, who was at the University of Madison, Wisconsin, has also contributed several lines that he had made along the same purpose in terms of having reporter lines there. So now you have a choice. You have an IPSC line or an ESC line. They're all in the same background, and they're well-characterized lines. They're all grown in the same media, and you can use them for your own screening effort. And if you decide you don't want to use those lines, you can go back and make your own reporter in your own line, but you'll have this comfort that you'll have faithful replication because the construct will work, and that it'll give you the same level of expression because it's in the same site with the same process. Okay. So I'm going to end here that I think cost is a big driver for all of this, even for looking at gene discovery or looking at drug discovery work. And you need to make sure that the screening opportunity, which is a low-hanging fruit, is not untapped, and that probably is the first way to go in terms of getting utility, and I gave you some examples of why that can be done. I think a crowdsourcing strategy can work. I gave you some examples of what's available, and all the data I showed you is from lines which are freely available, right? That's the important thing I want you to keep in mind. I'll skip the remaining part of the slide, and I'll end here and be happy to take questions. So we have time for some questions, I think. And if there are no questions, I just want to point this out to everybody.
Thank you, Mahendra, for a very illustrating talk. You mentioned you identify some compounds for Gaucher syndrome. Can you maybe comment on, after you went all through this pathway of having the cells screen compounds and you have identified the compounds, how long then it took to go into the clinical phase, so the last piece of the pathway?
So it all depends on the compound, right? If it's a known compound and it's part of the existing library of approved drugs, then you can be in the clinic in 6 months, right? If on the other hand, what you've gotten is a new compound or a new member of that family of compounds, then it's a new drug. And then it's going to take you 5 to 8 years to be able to do it. And you have to remember, for a new product, we don't have any data on its bioavailability, right? So you have to do all of that preclinical work, which you already have with this thing. And that's why a lot of people are screening in what people call not a really true high throughput large compound screen, but with the known compound library screens in the hope that they will be lucky and be able to get something that they can go to the clinic relatively quickly.
Do we have any other questions? Okay, so thank you very much again, Mahendra, for your presentation, and feel free to informally approach him for any questions. Thank you.
Thank you.